Welcome to the University of Pittsburgh Health and Explainable AI podcast, a collaborative initiative between the Health and Explainable AI HexAI Research Laboratory and the Department of Health Information Management at the School of Health and Rehabilitation Sciences, and the Computational Pathology and AI Center of Excellence, CPACE, at the University of Pittsburgh School of Medicine. I'm your host, Jordan Gass-Pooré, a health and science reporter. In this podcast series, we explore the transformative integration of responsible and explainable artificial intelligence into health informatics, clinical decision-making, and computational medicine. From reshaping diagnostic accuracy to enhancing patient care pathways, we'll highlight how AI is creating new bridges between researchers, clinicians, and healthcare innovators. Led by Ahmad P. Tafti, Houman Rashidi, and Liron Pantanowitz, the HexAI podcast is committed to democratizing knowledge around ethical, explainable, and clinically relevant AI. Through insightful conversations with domain experts, AI practitioners, and students, we'll spotlight the latest breakthroughs, discuss real-world applications, and unpack the challenges and opportunities that lie ahead in responsible AI in healthcare. So, whether you're a student, practitioner, researcher, or policymaker, this is your gateway to the future of AI-powered healthcare. Today, we're going to speak with Peter Maurer, Assistant Professor of Molecular Engineering in the University of Chicago Pritzker School of Molecular Engineering. Peter's a top researcher studying quantum sensing. On this podcast episode, we're going to talk about the future of pathology and laboratory medicine, and with Peter's help, we're going to look ahead to the quantum computing age. So Peter, it's an honor to have you on our podcast.
Thanks for hosting me.
All right. To get us started, would you like to introduce yourself? And next, can you give us an overview on the what and how of quantum sensing? For example, we often hear that quantum computers are incredibly delicate and susceptible to environmental noise. Can you explain how this very fragility is what makes quantum systems such powerful sensors? And then, could you walk us through a simplified example of how a quantum sensor works that might be relevant to pathology?
So as you said, I'm an assistant professor at the University of Chicago. I'm leading a quantum engineering lab. We are interested in developing new types of quantum sensors that can be used to probe the physical properties of biological systems at the nano and the micro scale. We have mainly two research thrusts in my lab. So the first thrust is aimed towards exploring the fundamental limits of quantum sensors in noisy, messy biological environments. And the second part is more translational, taking existing quantum sensing modalities and identifying biological and medical application, and then work with people in the life sciences to actually engineer our system for those applications. So you were asking what makes a quantum sensor so sensitive and how it is related to kind of the challenge in quantum computing, something that many of you probably heard of. So when building a quantum computer, one of the biggest challenge, if not the biggest challenge, is that quantum computers are very sensitive to the slightest perturbation from the environment. And so that has been the reason why it has been taking us so long just to scale from one to a couple of qubits. And so it's that high sensitivity that usually is seen in quantum computers as a problem. We take that as an advantage to develop better sensors that can probe very small perturbations, very small magnetic fields, electric field, temperature fluctuation, and then use that to learn something about nanoscale systems such as biological systems. What makes our approach a little bit different from quantum computers, because quantum computers are so sensitive to perturbation, they typically operate at cryogenic temperatures in ultra-high vacuums. So my lab and other groups around the world have developed quantum systems that actually can operate under ambient conditions and that are optically addressable, while at the same time maintaining this inherent quantum properties that a quantum computer has and its inherent sensitivity to perturbations in the environment. So you were asking about walking you through a simple example of how these quantum sensors may be relevant in pathology. So let me give you an example of something people have been talking about for the last maybe 15 years. So if you can measure with a quantum sensor magnetic fields very accurately, very precisely, you could use that to probe magnetic field fluctuations created by nuclear spins in nearby biomolecules, in nearby proteins. And this could enable a new type of nanoscale MRI or NMR spectroscopy that can provide us with chemical information in a not invasive way to probe signaling pathways like phosphorylation events in diseased and healthy states.
Thinking broadly about the future, can you offer any predictions on how pathology and laboratory medicine might potentially be impacted by advances in quantum sensing? What's your science fiction version of how the future might look?
Yeah, so the example I was mentioning before, these nanoscale NMR probes that can detect small structural changes in post-translational modification of proteins could be really a field opener in biological research, but also to study things like interactions of pharmaceuticals with GPCRs receptors, for example, or protein aggregation in neurological diseases like Alzheimer or Parkinson. So those I would say are kind of more on the far future technologies or applications that we are definitely working toward. But there are actually also some nearer term applications. And so particularly our quantum sensors can have very unique optical and spectral signatures that allow us to perform background-free measurements. And there are by now a couple of companies, one in Massachusetts and one in UK, that use this effect to perform very sensitive diagnostic tools that can detect multiplexed proteins at the sub-picogram milliliter volumes without the need to purify and wash and clean your specimen.
Switching to actual research project ideas that might inspire students, we love to ask our guests to share something they'd love to work on or that they'd love to see students try to tackle. What do you think?
So the things that I'm most excited to work on with students are projects I haven't thought of, projects that my students come up with. Having that said, I think I'm really excited at the moment in identifying problems that are really clinically relevant or relevant to biological, fundamental biological application. And so this is something where we need an interdisciplinary approach, where our group's knowledge in quantum technology and biophysics comes in, but then also the knowledge from people in other areas. So the things that I'm most excited about is problems where I can learn something new from another field.
At the University of Pittsburgh and Pitt's Computational Pathology and AI Center of Excellence, there's a lot of interest in the subject of synthetic data generation and uses of synthetic data to help train AI applications. What uses do you see for synthetic data in quantum sensing?
This is an amazing question. I think this is something that is completely underappreciated in quantum sensing. So at the moment, I and my colleagues have mainly been focusing on if we can measure magnetic fields very precisely, we can detect this and that nuclear spin species and learn something about structural changes or post-translational modification of the protein. But lately I've been talking to a friend of mine, Aaron Esther Khan, in immunoengineering here at U Chicago. And what he was actually proposing and what we worked out together is maybe our quantum sensors can be sensitive to chemical environments that we don't exactly know where these connections are, but we can see some effects on coherence. And maybe we can use our quantum sensors that can be sensitive to many different aspects of the environment to maybe learn something that some complex properties about the environment that would be missed if we just looked at the single type of measurement, just magnetic field or just electric field. So the sum of many different measurements done orchestrated together could provide more information than just the sum of each individual. And I think we really could connect with people in AI and learning about this kind of unstructured databases and things like this.
Another subject that Pitt is interested in is explainable AI, and Pitt's Health and Explainable AI Research Lab is carrying out cutting-edge work in the field. What are your views on explainability connected with quantum sensing? For example, how are you approaching explainability, and what have you learned?
Yeah, so let me give you an example, a very practical example from our own research. So we recently completed a project where we use quantum sensors in diamond nanoparticles that we can bring into macrophage-like cells. And we use these nanodiamonds to monitor small changes in our in the transition frequency of our qubit sensor. Okay, so what we found is that there is actually a correlation in this transition frequency with whether these macrophages get activated or are in kind of the resting phase. And so that was completely surprising to us. Once we identified this to be a reproducible mechanism, we started to dig into what's the physical origin and what's the microscopic mechanism that causes this. And we were actually kind of in for a surprise. What turned out is that these diamond nanoprobes, they are actually sensitive to the local electrochemical potential because on the surface you undergo optically induced redox reaction, but typically you don't realize that. And so what we found is that our qubit sensors in these nanodiamonds inside of a cell are actually really sensitive to slight changes in the charge environment and reconfiguration in the charge environment. And so it was kind of this coincidental finding through detecting that there were some overall changes in the qubit properties that led us to getting this more mechanistic physical understanding. And I think this is something where physicists and biologists really have a different approach to the world. As physicists, we say we can write down the equations that rule a system, and then we can apply some fancy mathematics, and we can make predictions and learn something surprising from that, and then we go into the lab and we test that, right? Versus biologists are kind of actually often the other way around. They say, we measured an effect, and now let's try to break down and figure out, break it down in the individual parts and figure out what is the cause of this. And we kind of applied here this biological approach, and we really had some very interesting insights into the physics of these qubits in nanomaterials inside of cells.
Lastly, Peter, we understand that you recently gave a presentation to Abbott employees on quantum sensing. The talk ran out of time, but we collected some follow-up questions that folks would have loved for you to speak on. Maybe we could challenge you to answer 4 of these questions that we found interesting. Quantum sensors overrate with extreme sensitivity, but biology is inherently noisy and heterogeneous. Could quantum technologies eventually help us redefine how we model biological complexity and not just measure it more precisely?
Yeah, that's a very good question. And a friend of mine here at the University of Chicago was actually making a very interesting statement related to this. He was saying, for a biologist, what's for a physicist noise is for a biologist a signal. And I think that applies here very well. So using our quantum sensor and looking at perturbations in the environment really provides insights into biological mechanisms in principle that would be very difficult to access with other technologies. So something we haven't done in the lab yet, but something one could envision, for example, our sensors can be made very sensitive to charge fluctuations. And so if you had one of these sensors nearby an ion channel in a neuron or in a heart cell or whatever, you could maybe detect the flow of individual ions through this channel. And so typically, if you looked at this, you would say, okay, that's just a noise, but it could maybe provide information to people in neuroscience that would be difficult to access with a patch clamp or something like that.
As quantum sensing moves towards practical biomedical applications, what are the biggest obstacles to scaling these techniques? Like from controlled lab setups to clinically relevant or in vivo systems?
Yeah, let me give two different answers to this same question. So the first one I would say is actually that this is a very interdisciplinary approach. There are questions on the quantum technology side, the quantum computing side that need to be worked out, but then we also need knowledge in biophysics and knowledge in medicine and molecular biology. And so bringing people together that can talk each other's language and team up to pursue a project like this has been a challenge. But here at U Chicago, we've actually had multiple mechanisms to create such a unique environment. And so one of them is we have a big NSF center, Cube, that is about a third of our members are from physics, a third from chemistry and engineering, and a third from actually biology and medicine. And we recently got funding for the first quantum medicine program at the Bergen Center here at U Chicago. And this will create even a bigger circle of clinicians and scientists working together to explore new directions in quantum biosensing. The second kind of answer would be on the technical side, and that's, I would say, sensitivity is still sometimes an issue. We would like to have ideally bigger sensitivity, but I think the bigger issue has more been interfacing quantum sensors with biological target systems. And that's something that my group has been very actively working on. And there has been over the last couple of years a lot of progress, and some of them came out of my lab, such as making diamond surfaces that can harbor qubits with very high sensitivity, completely biofunctional, to actually the point where we now are moving away from diamond and use fluorescent proteins as qubits for sensing. So these are all kind of new directions that just came out in the last couple of years or even months.
You covered some of this question already, but how do you envision coherent quantum sensors transforming diagnostic or therapeutic approaches in life molecule detection or brain machine interfacing?
Yeah. So I think one of the things that is still somewhat challenging with existing techniques is detecting small molecules in vivo, non-invasively. So no mass spec, no dissociation of your target system. And quantum sensors really offer here new possibilities through, for example, this NMR detection that I was mentioning, that could in principle detect the addition of a phosphate group to certain locations in a protein, or the interaction with a small pharmaceutical. To, in another direction, maybe detecting metabolites. That's something my lab is very interested in. We are now in the process of developing arrays on our quantum sensors that can detect dozens of different small molecules in very small sample volume. I think these kind of direction could have very direct clinical applications. And I try to not separate clinical and basic research applications because I think these two things go hand in hand. And certainly on both sides, there could be a big impact.
In your research interfacing biomolecules with quantum sensors, what are the key physical or biochemical challenges in maintaining quantum coherence in biological environments? And how does your work address these challenges?
Yeah, so up to very recently, people were thinking that diamond is the only system where you can kind of have a clean enough environment to get good qubit coherence at room temperature. And so what people have done is they kind of crushed off this diamond into nanodiamonds. And interestingly enough, if you stick this nanodiamond into living cells, into organisms like C. elegans or into tissue, the quantum properties remained maintained to some level. What is interesting is that in the last couple of months, there have been a few papers on as preprints become available, one actually from my lab and my colleagues here at U Chicago, where we show that even in a protein, you can get qubit properties maintained over a prolonged time. And so I think while it sounds very counterintuitive that you can maintain quantumness in biological systems, in this kind of noisy environment of biology, it turns out that you can actually. And so we are now exploring these things as sensors that are very biocompatible. Whether biology uses quantum for anything, that's a different question that I don't know. But we can definitely use quantum coherence to study biological systems.
Peter, I'm curious. How can outside partners like private sector companies and research labs at the University of Pittsburgh and elsewhere be of use to you and your research? And how do you envision that collaborations could work?
Yeah, I think there would be actually two ways that come to my mind where there would be great synergies. One is learning from you guys, where is the biggest need in industry for better sensors, for better measurement approach. The other thing is academic groups are in the business of publishing papers, of getting students to graduate, go for a postdoc, become academic leaders in their field and build up their own group. And that typically requires that you perform a proof of concept experiment, but it cannot be kind of you don't have the resources and the time to build turnkey system that can be used by other people. And I think national laboratories and industry could really come in there and help. And a good example is single molecule biophysics, right? Single molecule microscopy was developed. The first signal of individual molecules was seen in the early 90s by W.E. Maurer and other people at cryogenic temperatures. From there, it took some time, both scientific and engineering advances, to come up with super resolution microscopy. And then it still had to be turned into an actual microscope. And that required collaborations with the big microscopy companies. And I think we will see and require similar things on the quantum sensing side. So if there are companies that are interested in exploring new diagnostic devices, we would love to welcome them and have them be part of our discussions on what could be new directions to go to, and then also spin out our technology and have somebody to turn this into an actual device that can be used.
Peter, thanks again for joining us today. This is a short interview, and we'd love to hear more about your work. But we have to wrap up soon. However, before we do, would you like to leave us with a takeaway or give a shout out to your colleagues?
Yeah, sure. So for me, kind of the most exciting part is being part of this interdisciplinary interdisciplinary community, bringing together people from quantum technology, biology, biophysics, material science, to develop new type of sensing approaches that can be useful in the life sciences. And I think being in kind of this interdisciplinary environment at Chicago, in Illinois, in the US, is really something unique. And I'm really happy to be part of this.
Peter, again, it's been a huge pleasure speaking with you.
From the University of Pittsburgh, thanks everyone for tuning in and for following the series. Join us again on HexAI, where we explore the transformative integration of responsible and explainable artificial intelligence into health informatics, clinical decision-making, and computational medicine.